Good evening, everyone. I'm Tim Rogers, Chair of the Danforth Center Friends Committee. Welcome to the Danforth Center's virtual conversations. And we are thrilled to have two remarkable guests with us tonight. Dr. Jeffrey Gordon of Washington University in St. Louis and Danforth Center Principal Investigator, Dr. Becky Bart. Jeff and Becky are among the world's leading experts on the microbiome, and I'm really looking forward to hearing their stories and their perspectives this evening. But first, some gratitude. Thank you to our Friends Conversations Committee for organizing these programs. And thank you to our longtime media sponsors, St. Louis Public Radio and HEC Media. And thanks to you for being here tonight, especially those who are with us the first time. I'm sure you're going to want to come back. I'd like to encourage you to visit danforthcenter.org to learn more about the work of our scientists and the various contributing groups. For example, we have a great Young Friends organization, and coming in late September, they'll be hosting the Grow Challenge. This virtual fundraiser supports the Danforth Center's most crucial initiatives, and I hope you'll be able to join us. Later, you'll receive a brief email survey on tonight's program. We really depend on your comments and suggestions to keep making improvements to these programs. And now, our moderator for this evening, Senior Vice President for Compliance and Governance with the United Soybean Board, Darryl Chatman. Take it away, Darryl.
Good evening and welcome to Conversations. As a member of the Danforth Leadership Council, I'm excited to introduce tonight's panel. Dr. Jeffrey Gordon is known as the father of the microbiome. He is a world-renowned biologist and expert on the human gut microbiome. He holds the Dr. Robert J. Glazer Distinguished University Professorship at Washington University in St. Louis. Dr. Becky Bart is a principal investigator at the Danforth Center, where her lab works towards solving scientific mysteries for the benefit of farmers. The Bart lab specializes in the interactions between plants, microbes, and the environment. She is an expert on the microbiome of the soil. He is an expert on the microbiome inside our bodies. And as we will learn, these topics are related. And now let's begin our conversation with Jeff and Becky.
It's a pleasure to be here today and to talk to you about a project that is a collaboration between our colleagues at the International Center for Diarrheal Disease Research in Bangladesh and a group of very talented students and postdoc who work at the Center for Genome Sciences and Systems Biology at Washington University Medical School. Feeding our children. I'm going to take you on a journey in the next 10 minutes of discovery, trying to understand how to address a very vexing and pressing global health problem. That is childhood undernutrition. It's the leading cause of death of children under 5 years of age. A lot of epidemiological studies have shown that undernutrition isn't due to food insecurity alone. And current therapies have been able to reduce mortality, but they've had very limited success in overcoming the long-term consequences of undernutrition, which include persistent stunting, poor responses to vaccines, and neurodevelopmental abnormalities. So we're missing something, and what is it? So the hypothesis that we're pursuing is that the community of microbes, the microbiota in our gut, has a definable program of assembly that begins at birth, and that there is a disruption of this community assembly process that is causally related to undernutrition. A corollary to that hypothesis is that we have to have a more expansive view of human postnatal development. Includes one that encompasses our microbial cells and microbial genes, as well as our human cells and human genes. Healthy growth of infants and children is linked to healthy development of their microbiota. So how do we go about testing this hypothesis? First, we have to understand what is normal. So together with our colleagues in Bangladesh, we've studied birth cohorts, children who are followed monthly beginning at birth for the first 2 years and then subsequently the first 5 years of life with monthly sampling of their gut microbiota. There are technologies now that don't require us to culture the organisms in the gut community, but rather rely on DNA sequencing to determine who's there. These analyses of the developing communities of healthy children living in Bangladesh have identified a small group of covarying gut bacterial strains that describe a normal program of community development. Again, this is in healthy individuals. Remarkably, this group of organisms are also represented in the developing communities of healthy children living in disparate parts of the world. So when we think about human developmental biology, there's a shared program of assembly of our gut microbial communities. In addition, these types of analyses have shown that children who present with acute malnutrition during the first 2 years of life have a perturbation in assembly of their communities, leaving them with gut communities that appear younger than you would anticipate based on their chronological age. These defects in community assembly that we see in children who have acute malnutrition are not repaired by current nutritional interventions, none of which were designed with knowledge of or consideration of the developmental biology of the gut microbiota. So, what can we do? We first have to determine whether this immaturity is an effect of undernutrition or a cause. This test of a causality is conducted in the type of laboratory that's shown here in this slide. A gnotobiotic mouse facility where mice are reared under completely sterile conditions beginning at birth throughout the course of their lives. And we have the ability to introduce human gut microbial communities at specified points in their postnatal development and feed them the diets that are representatives of the diets of the donors of those microbial communities. With this capacity in hand, we can introduce microbial communities from children that are growing in a healthy manner, normally developing gut microbiota, as well as communities from children who have acute malnutrition, who have immature communities, and ask how much of the biological features of these children is transmissible to the recipient mice. So we're portraying the interaction between microbes, human microbes, and human diets in these animals. And what we see is that many of the features of undernutrition, children who have moderate acute malnutrition or severe acute malnutrition, can be transmitted to these recipient animals via their microbiota. And these features include impaired growth, muscle and bone, impaired metabolism, and impaired immune function. We can then inventory the microbes that have been transplanted into these mice and ask which microbes are associated with growth. These growth discriminatory strains become our therapeutic targets. They are underrepresented and presumably underperforming in the gut microbial communities of children with undernutrition. We're funded by the Bill and Melinda Gates Foundation, so at a very early stage in our discovery process, we have to think about issues of affordability, cultural acceptability, and scaling. So our approach to trying to find a way to repair the immature microbial communities of children with moderate acute malnutrition or severe acute malnutrition is to inventory foods that are consumed during a period of time that is present between when children cease exclusive milk feeding and when they are fully weaned. This is called the period of complementary feeding. So what we wanted to do is to go to Bangladesh to inventory the complementary foods that are culturally acceptable, affordable, and available, and try to identify those foods that have nutrients that are coveted by these underrepresented, underperforming, growth-associated organisms, and thus to be able to repair these communities. We wanted to incorporate these ingredients into what we call microbiota-directed complementary food prototypes and test their capacity to repair the microbiota in these living test tubes that we call gnotobiotic mice. So these mice were colonized by the immature communities of children with acute malnutrition, and we conduct our screens of these foods in these animals. These screens yielded MDCF, microbiota-directed complementary food prototypes, which we then advanced to human studies. I'm going to tell you briefly about a proof of concept study that we performed in Bangladesh involving 12 to 18-month-old children with moderate acute malnutrition who were wasted. the rate of weight gain was markedly below that of healthy children. We administered these lead microbiota-directed complementary food prototypes, specifically one that we call MDCF2, versus a standard nutritional intervention which was not designed with an eye towards the developmental biology of the microbiota or any evidence that it could repair these abnormalities that I just described in the microbiota of these children. It's called a ready-to-use supplementary food. So these different food were administered as supplements to children over a 3-month period of time. One group of 60 children getting MDCF2, another group of children getting this rice and lentil-based RUSF. During the course of this study, we sampled the microbiota of these children. We sampled their blood, measuring the levels of 5,000 different proteins that were important biomarkers and mediators of different aspects of growth. At the end of the period, we also were able to determine whether the growth of these children was affected by the intervention. Twice daily administration of MDCF2 produced a markedly greater rate of weight gain in these children than did RUSF, even though the caloric density of MDCF2 was 20% lower than that of RUSF. So we can pause and think about the implications of this set of observations. It isn't just calories that define nutritional value. And also, if we look at the 5,000 different plasma proteins that we measured, we see how profound the effect of microbiota repair are on so many different aspects of host biology, including mediators of bone growth, mediators of neurodevelopment, mediators of immune function. So we step back after a set of observations of this type and think about healthy growth of children. These types of studies imply that healthy growth is linked to our microbiota and that the activities of our gut microbial communities are felt well beyond the walls of the gut. This gives us a more encompassing view of human developmental biology, again, one that integrates both the contribution of our microbial genes and our human gene, and shows you, again, how pervasive the effects of these microbes are on our biology. It also gives us an opportunity to define wellness in a more comprehensive way. So we could think about the future in which children coming in for wellness checks have assays of the state of development of their gut microbial community. That there might be a window to intervene because this process of community assembly is largely completed in the first 2 years of life. Then when we see defects in community development, we might intervene, and those interventions of the type that I just described could have long-lived effects. There's also implications about policies related to complementary feeding practices. These decisions about what to feed our children at the time of cessation of exclusive milk feeding are questions that every family in the world asks itself. And I think with an idea of food intersecting with the microbiota, we in the coming years will have a better set of guidelines to nurture healthy development of the microbiota through the prescribed feeding of different sequences of food types. It also gives us an opportunity to think about how to identify the bioactive compounds in the foods that we use to treat the microbiota or repair the microbiota. And this is where our work intersects with Becky's work in the Danforth Plant Science Center. Microbes don't see food as labeled, asparagus or broccoli. They see the foods as a large and complex repertoire of chemical ingredients. And our ability to understand, as we are probing the structure-function relationships of MDCFs, what the bioactive compounds are in MDCFs, like MDCF2, allows us to develop future MDCFs that are adapted to different parts of the world where cultural traditions and the availability of foods vary. And finally, it's very important to see the consequences and generalizability of this type of approach to the treatment of undernutrition in children. For those reasons, we are now conducting trials in other countries. We're also looking at the effects of earlier interventions to see whether the effect size is greater, and also about the length of time that we treat. Can we promote greater repair of host biology if we treat for longer periods of time? We have a window of opportunity to be able to see how the nutritional value of food is influenced by our gut microbial communities. And I hope that that perspective will form bonds between the scientific programs that places, very special places like the Donald Danforth Plant Science Center is prosecuting, so that we really are able, in this time of population expansion, at this time of great challenges to sustainability, come up with more innovative ways of identifying affordable and more nutritious foods. Thank you.
All right, thank you, Jeff. Becky, why don't you go ahead and get started on your presentation?
All right, thanks a lot. Today it's my pleasure to try to tell you and communicate to you about the power of a healthy plant microbiome. Now, I'm going to focus in on one aspect of the plant microbiome, and that's the way that the microbiome is able to actually supply nutrients to plants. And even more specifically, I'm going to focus in on one of the most important nutrients, that is nitrogen. So nitrogen is the 7th most abundant element in the solar system. It's an essential building block of all life, and the majority of the nitrogen on Earth is in this gaseous form N2. That form contains an extremely strong triple bond, which makes it difficult for most organisms to use. We consider nitrogen one of the major limiting factors for plant productivity. However, in the early 1900s, two scientists, Fritz Haber and Carl Bosch, figured out how to convert that atmospheric nitrogen into ammonia. Ammonia is a form of nitrogen that's readily used by plants. And we call this industrial nitrogen fixation. And it is done in a factory like I'm showing you here on the slide. And this process has been so impactful, so widely used, that it's estimated that half the nitrogen in each of our bodies came through a factory like this. This is also a really energy-intensive process. It's estimated to consume 1% to 2% of the world's energy supply. And while this process has been incredibly impactful and has allowed us to feed billions of people, it also has come at some pretty significant costs. When farmers apply nitrogen to their fields, some of that runs off into our groundwater, into our rivers, lakes, and oceans. I'm showing you a picture of the Mississippi River and flowing into the Gulf of Mexico, and an algal bloom that has formed where that water is coming out. This process also degrades our soil and reduces biodiversity, and it's also a major contributor to climate change. So not only does it take a significant amount of fossil fuels to create that synthetic nitrogen, but when that excess nitrogen is applied to farmers' fields, some of it is returned to the atmosphere in the form of nitrous oxide. Nitrous oxide is a potent greenhouse gas. It's actually 300 times more potent than carbon dioxide. So in response to some of these environmental challenges, together with several of my colleagues at the Danforth Center, Chris Topp, Ivan Baxter, and Doug Allen, we recently established the Subterranean Influences on the Nitrogen Cycle, or SINC Center of Excellence. We're less than a year old, but we've already made a lot of progress, thanks in part to enthusiastic support from Phil and Sima Needleman. And the SINC Center has a really clear goal. It is to decrease or eliminate the need for chemical fertilizer while maintaining high plant yields. And we hope to accomplish this by promoting beneficial interactions with microbes and through optimized plant genetics. So I've told you a little bit about the problem, and I've introduced the team. Now let me tell you about the opportunity. It turns out that Earth comes with an endogenous nitrogen cycle. There are a lot of different microbes that live in the soil and associate with plants and are able to take that atmospheric nitrogen from the air and convert it into ammonia, which is that form that can be used by plants. Perhaps the most well-studied example of this type of symbiosis is with certain types of bacteria known as rhizobia that form intimate symbioses with members of the legume family, so soybeans or lupins. Over on the right side of the screen, I'm showing you a more recently observed example, which is an heirloom variety of maize that has these aerial roots coming out of its stem. And those aerial roots are covered in that gooey, thick mucilage. That mucilage is very carbon rich and is chocked full of nitrogen-fixing bacteria. So in both cases here, we have intimate interactions between a plant and a microbe and a very specific carbon-nitrogen exchange. Now, we believe that there's a lot of additional symbioses out there that have yet to be discovered, but we are now currently in a unique position where technology, especially phenotyping, next-generation sequencing, genome editing, and advanced modeling, is empowering plant science to do things that simply wasn't possible even a couple years ago. So let me tell you about some of the work that we've been doing over the last several years. This started with the simple question, can we develop ways to identify new symbiotic associations in the causal organisms? And we started with this very foundational hypothesis. Symbiosis, by definition, between plants and microbes can be observed as a positive correlation between plant fitness and microbe abundance. Now, summing up a lot of work in one single slide, let me show you how we tried to test that hypothesis. This is work that's been done with a whole lot of different scientists at different institutions and with funding from the Department of Energy, where we grew plants in the field. We collected their roots and used next-generation sequencing, just like Jeff was talking about, to identify all of the microbes that were associated with that plant. At the same time, we collected as many different plant health metrics as we could to really get a holistic view of the plant. And then we developed a computational pipeline to identify microbes whose abundance correlates with that plant fitness. Here I'm showing you just one example of a microbe that we observed, and you can see a really clear statistically significant positive correlation between plant health and the microbe abundance. And we've now applied this pipeline to many different data sets and have identified over 100 candidate beneficial microbes. The next phase of our work, we're really trying to complete the cycle and focus in on colonization and persistence. It's great to have identified these new potentially beneficial microbes, but if we can't get them to colonize and persist in our agricultural fields, then they can't perform that function. I can't tell you the answer to this yet because this is our active area of research, but I can tell you what we think is going on and how we're investigating this. So on the microbe side, we're gaining more and more evidence that microbe-microbe interactions are really important in determining the overall plant microbiome. On the plant side, we're gaining more and more evidence that the plant is not just an innocent bystander, it's actively cultivating these beneficial associations with microbes, and that some plants do that much better than others. To further interrogate these systems, we're using a number of different experimental systems that we have available at the Danforth Center. I've told you about culturing microbes and next-generation sequencing. We also have rapid seedling assays that we're able to screen through lots of microbes quickly. We've been fortunate to use a really unique facility at the Danforth Center, the Bellwether high-throughput phenotyping system. This allows us to conduct really highly replicated experiments in a very controlled way. More recently, we're starting to work with different technologies from Chris Topp's lab, like X-ray CT, and also larger mesocosm systems that are allowing us to really track nitrogen and carbon exchange and flow through these complex systems. Most recently, we're really excited to have established our first ever field research site out at Sella Farms, compliments of the Sella family for allowing us to develop this facility on their existing farm. So just to wrap up, I want to close with this Intergovernmental Panel on Climate Change 2021. I'm sure you all read all 4,000 pages of it, but if not, most of the major news organizations came out with the same take-homes. Climate change is real. Humans are to blame. Temperatures are rising. Weather is getting extreme. Seas will rise no matter what, and we're running out of time. To try to make that message perhaps a bit more palatable, they commissioned this nice piece of artwork to go along with it, and a quote from the artist. As we witness our planet transforming around us, we watch, listen, measure, respond. I really like this because we really, really do need to respond now. We are running out of time. And I don't want to end on a down note. I'd like to bring us back to the fact that there are many different puzzle pieces to fix these grand challenges, but I think plants and microbes hold a lot of potential, and I hope I've communicated that to you all at least a little bit today.
All right. I know I have some questions, and we have received some great audience questions, so let's jump into it. It's a really interesting presentation, so thank you guys so much. First off, can you tell us some of the some of the challenges facing your research as you move forward. We'll start with you, Jeff.
Thanks, Darryl. I like to think of the journey in several different parts. When we think about our gut microbial communities, how do we define normal? And is that definition of normal generalizable to other populations? And this requires, obviously, engagement with a particular population and the ability to follow, in our case, the development of a microbial community in groups of children over long periods of time. So the degree of devotion and trust that the mothers have for their children and the mothers have for their healthcare providers is enormous and makes this possible. Second, developing measuring tools for quantifying deviations from normal. We live in a very exciting time in this area of microbiome research, but it requires the application of high throughput technologies, the generation of very large data sets, and the capacity to use bioinformatic, computational biological tools to mine these data sets. That in turn requires a new generation of students who can do both experimental work and computational work. So once you have these measurements and you see variations between children, in this case with a disease, and normal, and the degree of deviation from normal correlating with the severity of disease, you have to do this test of causality. In this particular case, we use a living test tube, these mice, where we're able to take well-preserved microbial communities and transplant them into these animals to see how much of human biology can be ascribed to the microbes. But I think the greatest challenge is once you have preclinical proof of concept, to develop ways of manipulating a microbial community, repairing a microbial community that are affordable, culturally acceptable, and scalable. We're here today to think about food. We're thinking about food from a different perspective. The value of food, the nutritive value of food not being absolute, but rather a synthesis of the interaction between the chemical ingredients of food and microbes. And the ability to screen different food staples and try to find chemical entities that can be utilized by microbes, I think illustrates this marriage, this critical marriage between microbiome science and nutritional science. And I hope that that marriage is based on mutual respect and devotion, like all good marriages, and that it is sustainable. I think finally my comment is that really important to do rigorous clinical studies to get a very deep definition of the biological state of humans. In this case, we use measurements of 5,000 different proteins. When we repair a microbial community, to connect the dots between reconfigurations of different components of these communities and different aspects of the operations of human biological systems. Much like when you're trying to repair microbial communities that interact with root systems, to be able to get a very elaborate and comprehensive definition of the state of health and operation of a plant and the state of operations of a microbial community, and to connect that. So we look forward to determining the efficacy of this approach in different populations, the durability, and the long-term safety, which I hope we'll talk about later on.
Okay, all right, thank you. It sounds like the future is pretty bright. Despite those challenges. So Becky, can you tell us some of the challenges facing your work moving into the future?
Sure. I think one of the nice aspects of this conversations program is how many parallels there are between human microbiome work and plant microbiome work. Certainly I echo a lot of Jeff's comments. The sequencing costs, it's fun to look up if you have time, a graph of the price per base and how it has dropped dramatically over even the last 10 years. For sequencing, and yet we know that we're still missing so much. We're missing so much of the diversity that's out there, and our ability to understand how that complexity is really impacting the plants in our agricultural systems is still in its infancy. So it's a challenge, but also I think a place for real optimism. The only other thing I would say that's really pretty unique to plants is the challenges associated with studying roots. I'm very fortunate to collaborate with several people at the Danforth Center who really have a lot of expertise in that and novel imaging technologies, but I see that as another place where there's huge challenges, but also a lot of room for optimism. New technologies coming out of the medical field that are now being applied to plant systems and the like.
Darryl, I'd like to just make an additional comment and not be too verbose, but
Sure.
the similarities in the challenges and the technical approaches for surmounting those challenges when we study the symbiosis between microbes and plants and microbes in the gut in humans. And that is, and I'm going to get a little wonky on you, the capacity to sequence DNA and identify genes in microbial communities has accelerated greatly. But just knowing the genes that are present in a microbial community, and by the way, in our gut microbial community, there are at least 100-fold more genes than there are in our human genome, doesn't tell us how these communities operate. So one of the investments that the Donald Danforth Plant Science Center has made is in something called mass spec-based metabolomics. So it's the expression of genes in different microbial actors. What are the products of these genes? How do they transform molecules like molecules present in food or in soil? And how do these molecules actually serve as communication agents between microbes and the host? That's a huge challenge, but it's these technologies that are going to be essential to understand how these communities operate, not just their structure, but their function. And I think that's what really excites us as people who are living our lives in a world of microbes.
Okay. Well, Becky, can you talk about some of those technologies that may be developed in the future that can help with your research and moving your work forward?
Sure. So there's I completely echo what Jeff said about the limitations of just sequencing DNA, but it is still a very useful method for figuring out who's there. Most of the work that we've done so far has relied on short read sequencing, what we call it. It's where you take a genome that might be 1 million base pairs or 5 million base pairs or 20 gigabases and cut it up into little 100 base pair pieces and try to put it all back together and understand it. Some of the technology that's coming out now is what we call long read sequencing, and that technology is getting better and better, where instead of cutting your genome up into 100 base pair sequences, maybe you cut it up into 20,000 base pair sequences, and already you've been able to learn a lot more, and reconstructing those communities becomes much easier. There's also a lot of technologies I mentioned in my slides around genome editing. I think genome editing has allowed us to test hypotheses in a way that just wasn't possible before. There are some really specific challenges to doing genome editing in plants. We're doing it in my lab, we're doing it in a number of other labs at the Danforth Center, but there are some challenges there that I think need to be overcome and will allow us to test those hypotheses even faster.
Okay. So can you tell me, we're very proud. I'm from St. Louis, native St. Louis, and we are so proud to have the Danforth Sciences Center, Plant Sciences Center here. Can you tell us the advantages of doing this work in this region or in St. Louis specifically? Jeff?
About the Danforth Plant Science Center, or just this idea of food and the how to design food systems in the future that will produce products that have enhanced nutritional value. Of course, I think we're remarkably well situated here in St. Louis to form strategic alliances with one another. It's a place where people are kind, generous, lack affectation, and where collaborations are very easy to establish and sustain. I think that's really part of the culture of this place that we're in right now. It's a part of the culture of Washington University. I think the challenge, and one of the things that has been surmounted by the Danforth Plant Science Center is going from model systems, what you can do in a greenhouse, to what you can test in a field system. And we have the same thing. how much of our preclinical proof of concept that we develop in systems like those involving gnotobiotic mice I described can translate to human populations. It takes a lot of organization to be able to move from bench to bedside or bench to field, and I think you're seeing these things play out right now. I think that it's going to be very important for strategic alliances to be formed between those who practice microbiome research and those who are interested in nutrition. And I think the same thing goes with respect to agriculture, and realizing that the value, nutritional value of the foods that we consume is influenced by a consumer's microbial community. Just like the chemical landscape of a crop may be influenced by the microbes that a plant experiences in a particular ecosystem. We live in a world dominated by microbes. We operate with great humility and hope that understanding these relationships will really make this coming century one where humans will be able to flourish more.
Becky, can you talk about the regional advantage or the advantage of doing your work in St. Louis?
Yeah, there's a couple of things that come to mind, and obviously we're surrounded by agriculture here in St. Louis, so it's very easy to make the case of why this work is important and the potential benefits there, and to make that case strongly in a way that people will understand, I think. I also like what Jeff said about collaboration. So before I lived in St. Louis, I lived in Berkeley, and I remember when I first moved here, it felt like people would cross the street to come say hi to me that I'd never met before. It was a little alarming at first, but I think that sort of, I guess we call it the Midwest friendly attitude or whatever, actually has transcended into science as well. So for example, Jeff, I sent him an email a while ago, and he agreed to come and spend an entire morning sharing his ideas and his thoughts on this new Sync Center. I think that sort of spirit of friendliness, willingness to help out, collaboration is really pretty ripe here. It's good.
Okay, great. Now, I'm an attorney in my day job, so the question comes up of how are microbiome products regulated? Do you have some insight on that? I think that's a fairly interesting topic. Jeff, why don't you get
started? Could you say that a little louder, because
Yes. How are microbiome products regulated?
Yeah, that's a really, really good question. So I've talked about microbiome-directed foods. This is I'll call molten area about IP and regulation. A conventional food is defined as something that's consumed based on its organoleptic properties, aroma, taste, texture, and nutritive value. But if the nutritive value of food is influenced by the conversion of components of food by microbes, how do we factor in what are the nutritional contributions of food without considering our microbiota? So do we classify microbiota-directed foods as conventional foods? Do we and that'll be an interesting thing to consider in the future and in present. Are they medicinal foods? We're not trying to necessarily restore a particular set of nutrients that are missing, which is sort of a medicinal food. They don't really fit in the dietary supplement, and depending upon the claims, are they drugs or not? So I think that this is something that's going to be very important to try to work through. It's very critical that these foods be distributed generally. Yeah. So we want to try to make sure that that's possible, but at the same time, if Phil Needleman were on this stage, you have to make sure that efficacy is well established in different populations and the long-term safety. We're trying to change human developmental biology. At least from a microbial perspective, we're looking at the effects of the microbiota in various aspects of development, neurodevelopment, bone growth, etc. So if a child comes in and we're repairing the microbiota rather quickly, what's the long-term consequences of that? And I think we have to factor that in with regulation. So it's a great question. I hope you remain interested in this area, and I hope you contribute to this area.
Okay, all right. Well, great answer. Becky, what are your thoughts? Regulation.
Yeah, similar to human microbiome, I think we can divide our thinking on regulation into at least two halves. So the work I talked about where we're trying to think about the plant side, what role does the plant have in governing and recruiting a healthy microbiome? Those products, if we're so lucky as to come up with a product, will be regulated the same way all other crops are. There's a very established way of doing that with all sorts of safety checks and well-defined rules. The idea of applying a microbe that you have isolated from soil as a seed coat, that's a pretty well-established thing. I was reading the other day there's over 100 products already available to the farmers that are sort of microbial based. And so as long as it's something that has been isolated from nature and we're putting it back into nature, I think the path is fairly well trodden at this point. There's the idea of genetically engineering a microbe. I think that gets into some really pretty complicated area that needs a lot of consideration and discussion.
Okay. Are there any, oh, go ahead.
Just add, if I can say so. If we think in a transcendent way, will the selection of different cultivars, will the assessment of which cultivar to advance, will the decisions about how to cook food or process food be influenced based on deeper knowledge of the chemical landscape of crops and what the effects of all of these different parameters are on that? So I think we're going to move towards an era of much more personalized nutrition, but also I think that this work is really going to influence a lot of decision making in terms of food systems.
All right. Well, great answers, and I'm sure our audience really finds this topic interesting, and thank you for your comments. Okay, so we have time for one final question. Okay, so Becky, if successful, what is the impact that could theoretically be achieved by cultivating a healthy microbiome in agriculture?
Thank you for asking that. I think I left everybody on kind of a downer note there at the end with the climate change report. So let me emphasize that we know as scientists that a plant can get all of the nitrogen it needs through these associations with microbes. It just doesn't normally happen that way. So we have the tools out there to overcome this problem. We just need to put in the effort, the funding to fund the research, the creativity, and the hard work. We also know that agricultural soils and the agricultural system is responsible for about 10% of the climate change greenhouse gases emitted. So it's not the whole puzzle. I want to be really clear about that, but it's a really significant part, and that's what leaves me with at least some amount of optimism.
Okay, great. Well, this has been such an inspiring and hopeful conversation, but unfortunately we are out of time. I hope we will all come away from this evening with a greater appreciation of science and the role it can play in creating a better future. Thank you, Becky and Jeff, for sharing your work and your expertise with us. Before we conclude this conversations event, I'd like to tell you about another great Danforth Center event that is just around the corner. AgTech Next is a three-part series of expert perspectives aimed at tackling climate change. For just this year, AgTech Next is free. The first webinar is September 1st, so visit danforthcenter.org for more information. Once again, thank you to Dr. Bart, Dr. Gordon, the Friends Committee, our media sponsors, and to all of you watching, thank you for being with us, and good night, everyone.